I'm talking today about neuroinflammation in Parkinson's disease, and I will try to convince you that in fact it is playing a role in neurodegeneration in this disease. For most of the time, researchers thought that Parkinson's disease is a disease which is only affecting the neurons, but this is really oversimplified. Our brain is made of neurons, of course, but we have as many other cell types. This includes glial cells, the blood vessels, endothelial cells, and even immune cells coming from the periphery. So all these cells interact in the brain, and the non-neuronal cells also participate to the cascade of events leading to neurodegeneration. The first evidence that neuroinflammation plays a role in the pathophysiology of the disease came from epidemiological studies where healthy control subjects were analyzed, and those who took ibuprofen, in fact, have a lower risk to develop Parkinson's disease. This is not true for other non-steroidal anti-inflammatory drugs, but at least it's suggesting that the neuroinflammatory processes play a role in the development of the disease. Genetic evidence also supports a role of neuroinflammatory processes, and so-called GWAS study, who analyzed different genes and susceptibility genes, identify several loci in the genome which are involved, in fact, in the neuroinflammatory processes. And the first studies were done 15 years ago, and now many studies have confirmed this. So clearly, both from an environmental standpoint, from a genetic standpoint, the neuroinflammatory processes are involved in the pathophysiology. Then the next question is, which cells are really involved in this pathological process? And we have to remember our old textbook where we learned that, in fact, we have two different branches of immunity, one which is called the innate immunity, which is not responding to a given antigen, and the other one which is adaptive immunity, which is responding to a very specific trigger. And what we have shown with other people is that the innate immunity, namely the microglial cells, are in fact activated and play a role in the neuronal degeneration in Parkinson's disease. These cells are activated in the brain post-mortem of the patients, and in animal study, when you block this activation, you prevent the degeneration of the neurons. We have also shown that the lymphocytes, which are part of the adaptive immunity, penetrate from the periphery into the brain, and that a subset of lymphocytes, namely the CD4 lymphocytes, are playing a really deleterious role. And the group of David Sulzer in the US has shown that it is even a subset of lymphocytes, of CD4 lymphocytes, namely those who recognized the alpha-synuclein, which is a protein which deposits in the brain of the patient, which are the cells which are involved in the pathophysiological process. So on this basis, can we use that as a target to slow down the pathological process? This has been done by many groups of investigators, and the clinical trials, about 20 clinical trials, have been started on the basis of stopping the neuroinflammatory processes. But so far they were either negative or they are still ongoing, so the jury is still out. I think this is very interesting, but we have a major drawback with this type of approach, and which is that the phenotype of the cells, of the microglial cells, and we have studied that in animal models, is changing. It is changing during the course of neurodegeneration. First, these microglial cells play a role with with pro-inflammatory mediators, so they are playing a deleterious role, but when the disease evolves, some are producing anti-inflammatory compounds. And if this is really occurring in the brain of the patients, this means that we should use anti-inflammatory drugs at a very, very specific time window. And this makes it difficult. So I think this represents still a very, very interesting target, but we will need to have molecules that penetrate nicely into the brain and which can be delivered to specific brain regions at a very specific time.